After covering the fundamentals of apoptosis, in this video we explore the heart of the apoptosis process, the caspase cascade. This cascade is like an intricate self-destruct sequence for the cell, driven by a family of specialized enzymes called caspases. These caspases act as the executioners of apoptosis, dismantling the cell with precision. Caspases are synthesized in an inactive form, called procaspases, and they reside in the cytosol of nearly all cells. This inactive state is essential. It ensures that caspases are only activated when absolutely necessary, preventing unintended cell death. There are two main classes of apoptotic caspases: Initiator Caspases and Executioner Caspases. Initiator Caspases, like Caspase 8 and Caspase 9 in mammals, are the ones that kickstart apoptosis. They exist as inactive monomers until they receive a signal, often via a specific adapter protein complex. This signal brings multiple initiator caspase monomers close together. Once in close proximity, these monomers dimerize, activating themselves. Once active, the initiator caspases target their primary goal: executioner caspases. Executioner caspases, including Caspase 3, Caspase 6, and Caspase 7, handle the actual dismantling of the cell. Unlike initiators, these caspases exist as inactive dimers. Initiator caspases activate them by cleaving specific sites, turning them into active enzymes. This triggers the caspase cascade, where each active initiator can activate multiple executioner caspases, amplifying the apoptotic signal. Once active, executioner caspases go to work, targeting hundreds of proteins to carry out the clean and systematic dismantling of the cell. Caspase 6 cleaves nuclear lamins, breaking down the nuclear lamina and ensuring the nucleus disassembles irreversibly. Caspase 3 activates a DNA-degrading enzyme by cleaving its inhibitor. This allows the enzyme to chop up DNA, a hallmark of apoptosis. Executioner caspases also dismantle the actin cytoskeleton, leading to blebbing on the cell surface. They sever cell-cell adhesion proteins, detaching the cell from its neighbors to make engulfment easier. They even expose eat-me signals like phosphatidylserine on the cell's surface by inactivating and activating specific enzymes, ensuring phagocytic cells recognize the dying cell. The caspase cascade is a self-amplifying and irreversible process. Once initiated, the cell is fully committed to apoptosis. This ensures a swift and efficient dismantling of the cell, minimizing any risk of damage to surrounding tissues. In summary, the caspase cascade is a beautifully coordinated sequence that ensures apoptosis is not just efficient, but also highly controlled, protecting the body while maintaining tissue integrity. Apoptosis can be initiated through two main pathways: the extrinsic pathway, driven by external signals, and the intrinsic pathway activated by internal cues. Both pathways ultimately converge on the activation of the caspase cascade, orchestrating the cell's systematic dismantling. The extrinsic pathway begins when external signal proteins bind to death receptors on the cell surface. These receptors belong to the tumor necrosis factor, or TNF receptor family. They have three key domains: an extracellular ligand-binding domain, a single transmembrane domain, and an intracellular death domain, which initiates apoptosis. A classic example of this pathway involves the Fas death receptor and its ligand, Fas ligand, found on killer lymphocytes. When Fas ligand binds to Fas receptors, the receptors cluster together, exposing their death domains. This triggers the recruitment of an adapter protein, FAD or Fas-associated death domain. FAD then brings multiple inactive caspase 8 monomers together, forming the death-inducing signaling complex, or disc. Within the disc, caspase 8 is activated through dimerization. This initiates the caspase cascade, setting apoptosis in motion. In contrast, the intrinsic pathway is activated by internal signals, often in response to stress, such as DNA damage or developmental cues. This pathway relies on the release of proteins from the mitochondrial intermembrane space, most notably cytochrome C. Normally, cytochrome C plays a role in the electron transport chain. But once released into the cytosol, it binds to an adapter protein called APA-F1, or apoptotic protease activating factor 1. Together, cytochrome C and APA-F1 bind deoxy-ATP and assemble into a wheel-like structure known as the apoptosome. The apoptosome recruits inactive Caspase 9 monomers, which dimerize and activate within the complex. This triggers the Caspase cascade, leading to apoptosis. A critical step in the intrinsic pathway is the release of cytochrome C, controlled by the BCL2 family of proteins. These proteins regulate the mitochondrial outer membrane permeabilization, or MOP, determining whether apoptosis proceeds. There are three classes of BCL2 proteins. 1. Anti-apoptotic proteins like BCL2 and BCLXL, which block apoptosis by inhibiting pro-apoptotic effectors. 2. Pro-apoptotic effectors like BAC and BAX, which directly induce MOP. 3. BH3-only proteins, which promote apoptosis by either inhibiting anti-apoptotic proteins or activating pro-apoptotic effectors. The balance among these classes dictates whether cytochrome C is released and, ultimately, whether the cell lives or dies. Although distinct, the extrinsic and intrinsic pathways can collaborate to ensure effective apoptosis. For example, some cells rely on the intrinsic pathway to overcome inhibitors like XIAP, or X-linked inhibitor of apoptosis protein. The XIAP binds to and inhibits caspases, setting a threshold that must be overcome for apoptosis to proceed. During MOMP, mitochondria release anti-IAP proteins like Smac and OMI. These proteins bind XIAP, preventing it from blocking caspases and ensuring apoptosis completes successfully. In the end, both pathways converge on the activation of the caspase cascade, which dismantles the cell in a controlled and orderly manner. The choice of pathway depends on the nature of the apoptotic signal, allowing the cell to respond appropriately to a wide range of stimuli and stresses. Apoptosis ensures that cells are dismantled and removed without causing inflammation. A critical step in this process is the phagocytosis of apoptotic cells by neighboring cells, primarily macrophages in humans. This step prevents the leakage of cellular contents, which could otherwise trigger harmful inflammation. Apoptotic cells signal their readiness for removal by displaying specific "eat me" signals on their surface. The most important of these signals is the exposure of phosphatidylserine, or PS, on the outer leaflet of the plasma membrane. In healthy cells, PS is confined to the inner leaflet of the plasma membrane, maintained by a phospholipid flippase that actively transports it inward. During apoptosis, this distribution is disrupted by two caspase-dependent mechanisms. Executioner caspases inactivate the flippase, preventing PS from being transported back to the inner leaflet. And executioner caspases activate a phospholipid scramblase, which shuffles phospholipids nonspecifically between the inner and outer leaflets. The combined action of these two mechanisms ensures a rapid and robust accumulation of PS on the cell surface, providing an unmistakable "eat me" signal to phagocytes. This dual mechanism is vital. Without inactivating the flippase, PS would take much longer to accumulate. Without activating the scramblase, the flippase's activity would counteract the signal. Phagocytes recognize exposed PS on apoptotic cells with the help of soluble bridging proteins, which bind to PS and interact with receptors on the phagocyte surface. This triggers the engulfment process, leading to the apoptotic cell being safely digested. Interestingly, PS exposure can also occur during some forms of necrosis. However, in necrotic cells, The process is caspase independent and results from plasma membrane disruption rather than orderly signaling. Macrophages are highly selective. While they efficiently engulf apoptotic cells, they avoid healthy ones. This selectivity is due to "don't eat me" signals displayed on healthy cell surfaces. These signals interact with inhibitory receptors on macrophages, preventing accidental phagocytosis. Apoptosis is far from a passive process of cellular death. It is tightly regulated, particularly by extracellular survival factors. These factors play a critical role in maintaining tissue homeostasis by ensuring that cells persist only when and where they are needed. A fascinating example of this regulation can be seen during the development of the nervous system. Here, an excess of nerve cells is initially produced. These cells then compete for a limited supply of survival factors secreted by their target cells. Nerve cells that receive enough survival factors survive and thrive. Cells that fail to secure adequate survival factors undergo apoptosis. This competition ensures a precise match between the number of surviving neurons and the number of target cells they innervate, creating an optimized and functional nervous system. Survival factors typically operate by binding to cell surface receptors, activating intracellular signaling pathways that suppress the apoptotic program. Survival factors achieve this suppression through several mechanisms. 1. Stimulating the production of anti-apoptotic proteins. For example, survival factors can promote the synthesis of BCL2 or BCL-XL, members of the anti-apoptotic BCL2 protein family. 2. Inhibiting pro-apoptotic BH3-only proteins. Survival factors can inhibit proteins like BAD, which promote apoptosis. 3. Regulating receptor activity. In some developing neurons, survival factor receptors promote apoptosis when unoccupied. Binding of survival factors halts this pro-apoptotic activity, emphasizing the dual role of these receptors in life and death decisions. The dependence on survival factors is not limited to development. It also extends to adult tissues, where these factors regulate cell numbers and maintain tissue integrity. Regardless of the mechanism, the outcome is the same. Cell survival depends on the binding of survival factors to their receptors. This precise regulation ensures that apoptosis occurs only when necessary, preserving the delicate balance of cellular life and death within the organism. Apoptosis plays a crucial role in maintaining tissue homeostasis, and its dysregulation can contribute to various human diseases. Both excessive and insufficient apoptosis are implicated in serious health conditions. Excessive apoptosis leads to significant tissue loss in conditions like heart attacks and strokes. In heart attacks and strokes, initial cell death often occurs through necrosis due to a sudden interruption of blood supply. Even after blood flow is restored, surviving cells in the affected tissue can undergo apoptosis, exacerbating damage. Future therapies targeting both apoptotic and necrotic cell death pathways may reduce tissue loss and improve recovery outcomes. Insufficient apoptosis allows cells that should die to persist, contributing to cancer and autoimmune diseases. Many cancers evade apoptosis due to defects in regulatory mechanisms. For example, overexpression of the anti-apoptotic protein BCL2, often caused by chromosome translocations, prevents apoptosis in lymphocytes. This survival advantage promotes their accumulation and leads to lymphoma. Another key factor is the tumor suppressor protein p53, which promotes apoptosis in response to DNA damage. Mutations in the p53 gene, found in about 50% of human cancers, impair this response, enabling cancer cells to survive and proliferate with damaged DNA. This also reduces the effectiveness of anti-cancer drugs that depend on p53 to induce apoptosis. Insufficient apoptosis can also disrupt immune tolerance. Mutations in the Fas Death-receptor or its ligand impair the elimination of self-reactive lymphocytes, which attack the body's own tissues, leading to autoimmune diseases. Understanding apoptosis mechanisms has opened doors to promising therapeutic strategies. BH3 mimetic drugs, which mimic the action of pro-apoptotic BH3-only proteins, are showing success in cancer therapy. These drugs bind to anti-apoptotic proteins like BCL2 and BCLXL, inhibiting their function and promoting apoptosis in cancer cells that depend on them for survival. As research continues, therapies targeting apoptosis regulation may revolutionize treatments for cancer, autoimmune diseases, and conditions involving excessive cell death. Thanks so much for watching. If you enjoyed the video or found it helpful, give it a thumbs up. It really helps us out. Feel free to share it with your friends or anyone who might like it too. We'd love to hear what you think, so leave a comment below with your thoughts or any questions you've got. And hey, don't forget to subscribe and hit the notification bell so you never miss any of our new content. Your support seriously means the world to us. Thanks for being part of the community.